Hello students, thank you for watching my lecture Structure of Polypeptides and Proteins. The whole lecture series about this topic is meant to teach you how polypeptides and proteins can be synthetically accessed. However, before we can dive into the synthesis, we need to understand how polypeptides and proteins are built up and what are the important building blocks, and that I will discuss with you in the coming minutes. Peptides and proteins are biopolymers which consist of 21 different alpha amino acids that are connected via the famous peptide bonds or amide bonds. On the left-hand side in the scheme, you see the amino acid. The amino acid is shown here in green, and on the left, you see the amino group. On the right-hand side, you see the carboxylic acid group, and in the middle, at the carbon atom, you see a residue attached. That is where the 21 amino acids distinguish each other, and on this carbon atoms beside the side chain, there is usually also a hydrogen atom, which is not shown in the graphic here. On the right-hand side, you see a part of a protein where this green amino acid had been incorporated, and there it undergoes this peptide bond formation with the red amino acid that is shown right beside the green one, and in the circle you see the famous peptide bond and its respective structure. Proteins are structural and functional materials. Enzyme catalyze, for example, almost any chemical reactions. Hormones induce biochemical reactions. Antibodies are involved in the immune system. Receptors recognize signals within cells, and the whole process of signal transduction is also performed by proteins. Then proteins can recognize DNA, and therewith they are involved in regulation of gene expression. And peptides and proteins represent building blocks for nanostructures and important for the Max Planck School Matter to Life. They can be used as building blocks in artificial cells. Then we have a special class of proteins or polypeptides that are just peptides, so short sequences, and they contain only a few amino acids, but they can be biologically active and important. One example are peptide hormones, or another example are toxins that are present in different species and that can sometimes harm other species very severely. So what are the building blocks proteins are made of? These are the amino acids. We have two special ones. This is glycine, shown on the top left. It is special because it doesn't contain a side chain. The side chain in this case is a hydrogen atom, so it contains two hydrogen atoms at the C alpha position. On the right-hand side, we see the other special candidate, and that is proline. This is a cyclic amino acid, and the side chain, so to say, is fixed to the amine group, which makes this, or this structural feature makes proline also very special compared to the other amino acids. Something I need to add at this point is that besides the trivial name of the amino acids, there is usually an abbreviation used. If you, for example, discuss longer sequences, then it's very handy to use these abbreviations, and there's either a 3-letter code for each amino acid or even a single letter code that is then very special for each individual amino acid. For glycine, the 3-letter code is GLY, so GLY, or the 1-letter code is G, and for proline, the 3-letter code is PRO, and the single letter code is P. So all other amino acids that are present in proteins, they can be grouped via different criteria. Here I would like to use the classification. The first group shown on the left top here in the blue box are the side chains, or are the amino acids are summarized here, that have alkyl side chains. This is alanine, valine, leucine, and isoleucine. Then there are amino acids present in these 21 amino acids that contain aromatic rings. This is phenylalanine, tyrosine, and tryptophan. And finally, we have amino acids with side chains that are derived from carboxylic acids, and this is aspartate, asparagine, glutamate, and glutamine. Moreover, we have two more groups that need to be discussed. We had on the previous slides the one amino acid group that was derived from the ones that have a acid group as a side chain. Now we also have a group of amino acids that have a basic side chain, and these are lysine, arginine, and histidine. And finally, we have the group that contains multiple heteroatoms. This can be, for example, serine, carrying a hydroxy group with an oxygen as the heteroatom. The same heteroatom occurs in threonine, also carrying a hydroxy function. Then we have cysteine with a thiol, or a so-called sulfhydryl group present that involves sulfur. Then we have the analog to the cysteine containing a selenium atom that is called selenocysteine. And finally, we have methionine that contains a sulfur atom as well and is characterized by a thioether functionality. So when we now stitch the amino acids together to form a oligo or polypeptide, then we put them together in a certain sequence, and this sequence of amino acids we call the primary structure. Important when we write such structures down is that we start on the left-hand side with the amino group of the amino acid, and on the right-hand side we end up with the carboxylic acid, and these two ends are termed of the polypeptide are called the N-terminus or the C-terminus. Besides having a primary structure that does not say anything about the three-dimensional conformation of our polypeptide chain, we now need to discuss how the amino acids along this polypeptide backbone can be arranged in three-dimensional space. And first, what we want to look at, how do the three-dimensional structures look like of amino acids that are close related in the sequence. And this is called the secondary structure, and the secondary structure refers to the way in which segments of the peptide backbone orient into regular pattern. So due to the resonance delocalization of the CN bond that you see down in the middle here, the CN bond is shorter than conventional single bonds. The conformation of the peptide backbone is characterized by the 4 torsion angles that you see in the scheme on the right top. Due to the double bond character that I already have shown before, that is indicated here, the free rotation around the CN bond is drastically reduced. The rotational barrier is around 105 kilojoule per mole. So therefore, due to this partial double bond character, there only exist 2 rotamers, the trans and the cis form, which are shown in the green and the red box respectively. So the angle omega can only adopt 0 degrees or 180 degrees. Due to the fact that the energy or the trans rotamer is 8 kilojoule per mole more stable than the cis form, it is found in most of the peptides that not do contain proline. In proline, the case is a bit different. In this case, the trans form and the cis form are energetically lower together, and in this case, the trans form is only 4 times more stable than the cis form, and that means in proteins you also find proline in a cis configuration. Let's move now and look what different structures can be adopted according to these 2 angles. So the angle omega is fixed, and now we need to concentrate on the 2 most important angles for the adoption of secondary structures, and this is Psi and Phi that are shown on the figure on the right. Most of these combinations of Psi and Phi are not allowed because of steric collisions of the side chains that are shown here with the purple balls with the backbone. It is straightforward to calculate all allowed angles, and possible combinations are shown in the Ramachandran plot that is shown on the left-hand side here. In this Ramachandran plot, you see 3 allowed regions. One of the regions expresses the angle space of the beta strands, and the green and the purple box, they indicate the left-handed alpha helices and the right-handed alpha helices. And what is not shown in the diagram is the angles that are allowed to be adopted by proline. Their phi is to be minus 60 degrees. That has to do with the connection to the amine. And finally, glycine allows much more angles to be adopted because glycine does not contain any side chain except the hydrogen atom that is placed there, and that is much smaller than any of the other side chains of the other 20 amino acids. So in general, we distinguish between 2 secondary structures this is on the one hand the alpha helix, and that is on the other hand the beta sheet. And now I would like to discuss with you the alpha helix first. The alpha helix is characterized by a helical turn that consists of 3.6 amino acids. In the ideal case, phi adopts an angle of minus 57 degrees and psi adopts an angle of minus 47 degrees. The whole helix structure is held together by hydrogen bonds. Only the last amino acids and the termini are not involved in the hydrogen bond network that stabilizes the helix. The hydrogen bonds that are formed are so the C-terminal NH group, hydrogen bonds with the carbonyl group at the N-terminal position, and within the hydrogen bonds, 4 amino acids are in between, and then the hydrogen bond, so the one hydrogen bond gives stabilization to 4 amino acids that are in between. And these 4 amino acids, in these hydrogen bonds, a ring of 13 atoms is formed. I recommend to you that you take a closer look at the picture on the left, and you do it for yourself. So you look for the NH in the helix, and then you count along the helical chain to find this ring of 13 amino acids, and then you can easily follow the statements that are made on the right hand side. Besides the classical alpha helix that is indicated on the picture on the right hand side, there are other helices, and this is the 310 helix and the 4.460 helix, or also named the pi helix. These helices are less abundant. The question is, what does the 3 and the index 10 means, and the 4.4 and the index 16? 3 means the number of amino acids per turn, and the index 10, that is the number of atoms involved in the ring, including the hydrogen bond formation. For the pi helix, it's the same. In this case, the pi helix contains 4.4 amino acids per helical turn, and the hydrogen bonds involve a ring of 16 atoms. Amino acids that are compatible with the helical structure are alanine, valine, leucine, phenylalanine, tryptophan, methionine, histidine, and glutamine. Proline is not compatible with the helical structure. Let's move now to the beta sheets. Beta sheets can be either formed by parallel strands of peptides, or they can form by antiparallel strands of peptides. And again, the beta sheets, they are stabilized again by a network of hydrogen bonds between 2 adjacent strands. If you want to understand the structure of the beta sheets better, then have a look at the picture on the right hand side. What you see is that you have a kind of roof-like structure that moves like this, and when you look at this roof-like structure, then always at the top of the roof, the side chains that are connected to the alpha carbon atom of the amino acid, they perpendicular, let's say, to the plane of the roof, they stick out either on this side or on the other side of the beta sheets. The beta sheets are usually 5 to 10 residues long, and phi and psi are within the broad structural allowed region of the Ramachandran plot. That's the upper left quadrant. And in the beta sheet, the amino acids that you often find are the ones with beta substitution. Rarely proline and glycine are found within the beta sheets. Now we come from the secondary structure to the tertiary structure, and the tertiary structure refers to the way how the entire protein molecule coils into an overall three-dimensional shape and describes spatial orientation of amino acids that are far in sequence. In the secondary structure, you have seen how amino acids orient in three-dimensional space that are very close to each other. However, the tertiary structure tells you how the entire polypeptide chains folds into a three-dimensional geometry. And this three-dimensional geometry of this polypeptide chain somehow needs to be fixed in space. And this is realized by different interactions that stabilize the tertiary structure that can be, on the one hand, covalent bonds. In this case, an example is, for example, disulfide bridges. Then, like in the secondary structure, hydrogen bonds can induce or fix the tertiary structure. Then opposite charges, meaning ionic bonds or salt bridges, can adjust the polypeptide backbone in space, or this can be realized by hydrophobic interactions of the interplay between two hydrophobic amino acid residues. Probably also important to note at this point is that these four different interactions are characterized by different bond strengths. The strongest bond is the covalent bond, in this case the disulfide bridge. Then hydrogen bonds have a bond strength of around 6 to 40 kilojoule per mole. The salt bridges are 40 to 85 kilojoule per mole, and the smallest interaction energy with 1 to 4 kilojoule per mole are the hydrophobic interactions. Finally, now we have another hierarchical level within protein structures, and these are the quaternary structures. In this case, it is described how either the same polypeptide chain, the same folded polypeptide chain, interacts with others to form a multi-enzyme complex, or how different polypeptide chains arrange in three-dimensional space to each other. This is the quaternary structure, and this is shown here on the right-hand side of the slide that you see. In this case, hemoglobin has been chosen as an example, and this consists of 4 protein units that arrange into the characteristic structure into the multi-protein complex that is able to store oxygen, and which is a major component of our blood. With this slide that gives you an overview about the primary structure, the secondary structure, the tertiary structure, and finally the quaternary structure, I would like to end my presentation here, and I thank you very much for your attention.